presentation will be given by the Dr. Higgins from Jeju, UK and New York, USA, and the title of her talk will be Microenvironment Reprogramming of Human Dermal Papilla Cells by Spheroid Culture Restores Their Capacity for Instructing Hair Neogenesis. Go ahead, please.
Firstly, thank you to the organisers for inviting me to speak. And I don't really have to put this slide in this talk, not for this audience, but it's one I like to use first because it enables simplification of what is quite a complex structure. So this is a macroscopic image of occipital scalp skin. And you can see on the top you have the epidermis, which is the skin barrier. Beneath this you have the dermis, which contains dermal fibroblasts among other cell types. And beneath this you have dermal adipose tissue. You can see that sebaceous glands are associated with the hair follicles, while hair fibers exit the skin surface. Hair follicles, which are predominantly epithelial in nature, actually run continuous with the epidermis and then protrude down through the dermis, down into the dermal adipose tissue. At the base of the hair follicle is this end bulb region here, which contains epithelial matrix cells and also mesenchymal dermal papilla cells, which I'll be talking about during my talk today. So hair follicles form through a process known as secondary induction, and the initiating signal for this process comes from the unipotent dermis. It's thought perhaps it's a wind signal, although its origin is unknown. So in response to this dermal signal, the epidermis starts thickening and forms a placode. A signal from the placode down into the dermis causes these cells to move together and form the dermal condensate. Now, the dermal condensate contains cells that are double the density of the cells within the interfollicular dermis. And when they move into this condensate, they start acquiring a condensate fate. The cells in the condensate signal back up to the placode, causing proliferation, and these cells then push down and invade into the dermis, first forming the hair germ, next the hair peg, and eventually the hair follicle. And you can see that as this process is occurring, the hair follicle starts to engulf these condensate cells, and they become known as the dermal papilla, which persists in the adult hair follicle and is critical for hair follicle cycling. So why am I so interested in dermal papillae? Well, about 30 years ago, Colin Yehoda showed that you can take rat whisker follicles, isolate the dermal papillae, and then place these cells into inductive assays where they're capable of instructing hair growth or hair fiber growth in recipient epithelium. You can also take these cells and place them into culture, and at low passages in culture, these rodent dermal papilla cells retain their instructive capacity and they can instruct hair growth in recipient epithelium. At higher passages, though, the rodent dermal papilla cells lose this capacity. But why is this interesting? If we could harness the instructive properties of human dermal papilla cells, we could envisage growing hair follicles in situations where there currently are none, such as in androgenetic alopecia or a skin graft or a burn. And I say in skin grafts because current skin models used to treat burns or non-healing wounds are devoid of hair follicles, and they don't represent the complexity of human skin at all. They don't have hair follicles, nerves, or blood vessels, and they're more like living bandages rather than functional skins. And so establishment of a hair follicle within a skin graft would be the first step towards making a functional skin. And so for many years now, researchers have been trying to demonstrate that human dermal papilla cells retain the same instructive properties as their rodent counterparts. And you can isolate dermal papilla cells, this is the papilla, this is the sheath, from scalp skin biopsies. You can place these into instructive assays where they are capable of instructing hair growth, but as soon as you put them into culture, the human dermal papilla cells quickly lose their instructive potential, and limited hair follicle induction has actually been demonstrated using human dermal papilla cells. And so there's a great disparity between species. At one end of the spectrum you have intact rodent DP, which are really the gold standard for induction, whereas at the other end of the spectrum you have your cultured dermal papilla cells, human dermal papilla cells. And so we wanted to understand why this was, and we obtained occipital scalp biopsies from 3 male donors. We isolated the intact dermal papillae and also established cell cultures from these biopsies. Then we isolated RNA from the intact papillae, cells at P0, which is the explant stage of cell culture, when the papillae is collapsed, but prior to trypsinization, and then cells at P1, P3, and P5, which have been passaged to divide them and expand them. From this RNA, we generated biotin-labeled cRNA and hybridized this onto Affymetrix gene chips for analysis. We then used a commercially available software called GeneSpring to identify transcripts that were both significantly and differentially regulated between the intact papillae and cells at subsequent stages in culture from the 3 male donors. And so this graph here shows every transcript that was identified in this manner that is significantly and differentially regulated between intact papillae and cells in culture. And you can see that there's a really dramatic change, not when the cells are trypsinized, but as soon as they hit the plastic, as soon as they go from a 3-dimensional to a 2-dimensional environment. And so the most significant changes in our papilla transcriptome occurred after a very brief period in culture, and we started to look back at the literature to try and understand this observation. And one thing that became very apparent to us that hadn't perhaps been so obvious before was the propensity of cells in culture to aggregate. So rodent dermal papilla cells, which are inductive, spontaneously aggregate very frequently, and they form these super aggregates here, as you can see on the right-hand side. Whereas human dermal papilla cells, which are not inductive, never spontaneously aggregate. Sometimes you do see aggregation, but it's more like this. I personally have never seen anything like this with a human dermal papilla cell culture. And I should say these were all cultured in just regular DMEM and 21% oxygen. And so this really pointed to the importance of aggregative behavior in dermal papilla culture and pointed to the importance of a 3-dimensional environment, inviting us to try a very simple experimental reversal strategy, which was we needed to promote aggregation within our human dermal papilla cultures and see whether this could promote their inductive capacity. So we grew human dermal papilla cells in hanging drop cultures, 3,000 cells, and we formed dermal spheroids, which are 150 microns in diameter. And actually, in vitro, human dermal papilla cells don't proliferate. They also don't express alpha-smooth muscle actin, but as soon as you put them into culture to grow them, they of course start proliferating. And they also switch on expression of alpha SMA, which is a well-known marker of dermal papilla cells in vitro. However, when we grew dermal papilla cells in hanging drops to form these dermal spheroids, they quickly stopped dividing, and after about 30 hours, they also switched off alpha-smooth muscle actin expression, which was quite encouraging. So we then wanted to know, well, are our human dermal papilla cells, when grown as spheroids, inductive? And there's a lot of different assays out there that you can use to test the instructive capacity of dermal cells under question. And these all vary in terms of their stringency or the challenge that they pose to the dermal papilla cells. But one thing that they all have in common is that they all assess the capacity of dermal cells to instruct hair growth in rodent epithelium, whether it be cells or intact skin. It's always rodent. And so we wanted to establish a human-to-human assay to ask, do our human spheroids instruct hair growth in human skin? So we obtained non-hairy human skin. We used disphase to separate the epidermis and dermis, and onto the exposed dermis we placed either intact dermal papillae as a positive control, cells cultured in 2D regular culture conditions, and cells cultured in hanging drops to form dermal spheroids. We then placed back over the epidermis, a bit like a sandwich, and placed this onto the back of a mouse for 6 weeks. The mice were bandaged, and at the end of 6 weeks we removed the grafts to assess them for the presence of hair follicles. And sure enough, the dermal papilla spheroids are capable of inducing de novo hair follicles in the non-hairy human tissue, and this is a couple of examples you can see here with the newly formed dermal papilla and sometimes a little bit of inner root sheath. Follicles were also observed when intact papillae were placed between the non-hairy human skin, which was our positive control. They were not observed when 2-dimensional culture suspensions were used, nor when no cells were used. Our induced follicles contained all human cells, as you would hope, since it was a human-to-human assay, and they also had a newly formed basement membrane. You can also see that they were alkaline phosphatase, and actually this extended in a sort of halo around the hair follicle. So all in all, we used 7 different donors to assess the instructive capacity of dermal papilla spheroids, and we only got hair induction with 5 of the donors. And actually that was induction of a hair follicle we only saw external hair growth from 2 of the donors, not all 5. So this does point to a limitation in our assay, that we're only getting a partial restoration of inductivity. And we don't know why this is. It could be because the epidermis isn't competent and capable of responding, since it is a non-hairy epidermis. Or it could be something intrinsic to the dermal papilla cells themselves. And so to try and understand the partial restoration of inductivity in our dermal spheroids, we then completed Affymetrix profiling on 48-hour-old spheroids from the same 3 donors that the original microarray analysis was performed on. And you can see we then performed unsupervised hierarchical clustering on the entire genome signatures of these Affy profiles, and you can see that the freshly isolated dermal papillae fall in their own little arm here. Cells cultured in 2D fall in a culture arm of this hierarchical plot here, as marked in green, and their dermal spheroids, while falling in a culture arm of the plot, do fall independently of the other cells in culture. A bit loud. We then performed a correlation coefficient analysis, and the average correlation coefficient between intact DP and cells in 2-dimensional culture was 0.42. Each donor is shown here, and this correlation is the light blue one. While between intact papillae and dermal spheroids, which is the dark blue, the average correlation coefficient was 0.56. So there was a small increase in correlation between papilla cells when grown as spheroids comparative to intact DP. And this small increase in correlation equates to a 22% restoration of the intact dermal papilla signature when dermal papilla cells are grown as spheroids. So every line on this graph again represents a transcript that either decreases or increases in expression in culture by culture, but then 22% of these transcripts are restored again when you grow the cells as spheroids. But this does raise the question, are we establishing something that's representative of a condensate, or a dermal papilla, or perhaps a hybrid of something in between? We're not really sure. So there's a lot of different ways that you can use to analyze your data set, and this is one that I particularly like because it enables you to visually represent your data. And it's called JEDI. It was developed in Don Ingber's lab at Harvard. And essentially every square here represents one of the different Affymetrix data sets that we were looking at. Across the top we have the 3 different donors, and down the rows we have either intact DP, cells at P0, P1, P3, P5, and at the bottom you have dermal spheroids. And then within each of these squares, there are actually 650 little squares on a 25 by 26 grid. And what JEDI does is it groups transcripts together into the same little node on these grids if they share similar temporal expression profiles across the entire data set. So then you can go in and find areas that are of interest to you, such as this area here that contains nodes with transcripts that are expressed at very low levels in intact DP, increase in expression in culture, and then go back down again in dermal spheroids. Or on the other side, you have genes that are expressed at high levels in intact DP, decrease in expression in culture, and go up again in dermal spheroids. And so we went in and we identified 4 of these metamosaic areas that were of interest for us. And I've already described T1 and T3, which represent the 22% of transcripts that we identified as restored. And then we also have T2 and T4, which contain transcripts that either increase or decrease in expression in culture, but are not restored when you grow the cells in dermal spheroids. So depending on what you're interested in, you should analyze the different territories. For example, if you're interested in the mesenchymal response during mesenchymal epithelial interactions, then you might want to analyze territories T2 or T4, which are the transcripts that were not restored by spheroid culture, because obviously that is something that we are not replicating in this model. I personally am very interested in analyzing territory 3, which is the transcripts that are restored by spheroid culture. And I think that analysis of T3 could give us insight into genes required for aggregation or condensation, perhaps migratory genes or adhesive genes, or maybe even condensate fate. And there's lots of different ways that you can then go in and analyze your different territories, and I've just got one example of this here. In collaboration with Andre Calfano's lab at Columbia, we reverse engineered a skin transcriptome using an algorithm called Arachne. And what Arachne does is it takes transcription factors and it puts them in a hierarchical plot. So you get master regulators of all the genes within the skin. And you can then use Arachne to interrogate your gene sets of interest or your territories of interest to identify master regulators of those territories. And this is an example of our territory 3 that was interrogated with Arachne. And you can see that Arachne predicts there are 4 transcription factors, as marked by the yellow nodes, that regulate the entire regulon, so transcripts that were present within T3. So another question is, what enables a condensate or an aggregation to actually acquire its inductive identity? Is it autonomous or is it conditional? Is it density dependent? And so one thing that we decided to do was to analyze the capacity of fibroblast spheroids to also induce hair growth. And so we took fibroblasts from the same donors of the dermal papilla spheroids who were inductive. We grew these in hanging drops to form dermal spheroids. These also contained 3,000 cells, and they were approximately the same size as the spheroids formed from the dermal papilla cells, although density is obviously something we can't control. We used our human-to-human assay to assess their instructive capacity, and we never saw any hair growth when we implanted dermal fibroblast spheroids. So that indicates that spheroid culture is necessary, but it's not sufficient for hair growth, and that the dermal papilla cells must have some molecular memory that is reactivated by 3-dimensional culture. And so if it's not density dependent, this begs the question, well, can fibroblasts become condensate cells if they obtain the correct conditional signals and replicating what would occur during development? Or perhaps can fibroblast cells be reprogrammed to a papilla fate using master regulators? But I believe this is something that Carlos Clavel will talk about in his talk tomorrow. And so in conclusion, I've shown you that human dermal papilla cells lose their capacity to induce hair growth when you remove them from the follicle and grow them in culture. By growing dermal papilla cells in spheroid cultures to mimic condensation or aggregation, we observe a partial restoration of the hair inductive capacity. In dermal papilla spheroid cultures, we observed a 22% restoration of the intact papilla signature, and genes whose expression is restored by spheroid culture, those in T3, may give us insight into the aggregative processes, such as cell migration or cell adhesion. Aggregation of dermal cells is necessary but not sufficient for hair induction. And lastly, I'd just like to thank Angela Cristiano. This work was done when I was a postdoc in her lab, and a lot of it was done in collaboration with Colin Yehoda in his lab. And lastly, thank you to Dr. Robert Bernstein at Bernstein Medical, who provided us skin for this study. And with that, I'll take any questions. Thank you.
Thank you very much for your wonderful talk. Any questions, comments? Yes, please.
That's clear. Did you try dermal shift fibroblast?
We did not, no, but I think they would.
The upper or lower dermal shift?
No, we established cell cultures, but I never had time to go down that route. But I think that would actually be quite a promising avenue to go down.
Jeju.
That's very nice. You had 3 main donors, is that right?
For the microarray, yes.
Yeah. So did you use the cells from those donors in the assay with the...
No, we didn't. We didn't have enough cells left, so the 7 donors that we used for the assay were a different 7 donors.
So you never used the ones that you used for the array, you never used those cells in the functional assay?
No, we didn't have enough cells, unfortunately.
Any questions? If not, let's move on, and I will pass the chair to Chris.